Hello. Thanks for tuning in to my talk. My name is Matthew Hasenjager. I'm a postdoctoral fellow at the University of Tennessee, Knoxville. But today I'm going to share with you some work that I did as part of my first postdoctoral position at Royal Holloway, University of London, exploring the transmission of foraging information through honeybee colonies. And before I begin, I just want to say thank you again to the organizers of the symposium for inviting me to participate and putting together a really great series of talks. I hope you enjoy them as much as I have. Social transmission is a process by which novel behaviors or information spread from knowledgeable individuals to uninformed individuals as a result of social learning. So, for example, here we see these monkeys, where this older individual perhaps is very proficient in a technique for opening up this fruit, and these two younger individuals may, by observing this older individual, be more likely to acquire this technique themselves. And when studying social transmission, social network analysis has really been an invaluable set of tools because it lets us represent potential pathways of transmission within a population. So for example, if we had this network here and where the nodes are individuals and the connections between nodes represent some form of social relationship, then we might, on the basis of this network, make some predictions like those individuals that are more centrally located in the network may enjoy enhanced access to social information, may be more likely or more rapidly pick up novel behaviors that are being spread through this population, as opposed or as compared to individuals that are located on the periphery of this network. However, many networks used in behavioral ecology employ just a single form of connection, where maybe we're using a single proxy measure, such as the amount of time two individuals are found near one another, to represent the likelihood for social information to spread between them, or we may collapse multiple forms of interaction into a single form of relationship. But this ignores the possibility that information may spread at different rates based on different ways that individuals may interact, or based on different ways that individual pairs may differ in the nature of the relationship with one another. So, for example, individuals may differ in the nature of the relationship they share with others. So, some relationships may be dominated by agonistic interactions, whereas others may be more affiliative in nature. And it may be that information or behaviors transmit more readily across certain forms of relationships than others. Likewise, individuals may interact across multiple modalities, whether that be visual, auditory, or chemical. Or the likelihood that transmission will occur may be dependent on the characteristics of knowledgeable individuals or the individuals that are observing them. So, for example, dominance may be a very salient feature, where individuals may be more likely to learn from dominant individuals if they tend to spend a lot of time observing those individuals and paying attention to where they are and what they're doing. Or it may be that, for example, the personality types or behavioral types within a group may shape how transmission occurs, where individuals may be more likely to interact or learn from certain behavioral types than others. The point being that given the variety of ways in which animals interact and given how relationships can vary in their quality, we may want to employ techniques that let us explicitly study the relative rates of transmission across these different forms of social connectivity, rather than simply aggregating them into a single measure of social connectiveness. One system that's an especially good example of this is the honeybee, where honeybees have evolved a suite of behaviors that allow them to effectively share information with one another and coordinate their activities to allow the colony to function effectively as a more or less cohesive entity. So, for example, honeybees share information about foraging locations using waggle dances. I'll talk a lot more about that later on. Or they employ stop signals as a form of negative feedback that can shut down the production of social information being produced by other individuals. They employ shaking signals and tremble dances, which may act as modulatory signals, signals that change the receptivity of the recipient of that signal in terms of their likelihood of responding to other signals that they might encounter in the hive. Honeybees also are adept at acquiring olfactory information, such as through trophallaxis, where they're sharing nectar samples with one another, or through antination, where they can learn about the scent of resources their nestmates have been foraging on, or the wide variety of pheromones that honeybees produce to share information broadly with others in the hive. So when we're interested in understanding how information is spreading or transmitting within a honeybee colony, we often need to adopt a multi-layer perspective to understand how multiple forms of interaction might be guiding that spread. So today I particularly want to focus on the context of foraging. So honeybee foragers, their main role is to fly out and collect nectar and pollen from flowers, which respectively serve as the main form of carbohydrates and protein for the colony. And so honeybee foragers ideally want to be able to effectively and efficiently locate foraging locations, especially ones that are relatively profitable. And so I'm interested in understanding how different forms of social interaction in the colony help bees to decide when to forage and how they use these different forms of social information to help them locate profitable foraging patches. And perhaps the best known honeybee signaling behavior is perhaps the waggle dance. This, you can see a bee here performing a waggle dance, the one in green and white. You can see it's moving in this kind of figure eight pattern across the comb. And what the waggle dance allows is for bees to be able to share the distance and direction of profitable resources that they've located out in their broader environment with nestmates back in the colony at home. So in other words, this bee could fly out, locate a site 5 kilometers away from the colony, come back to the hive, share the coordinates of that resource, that food site, with their nestmates who can then fly out and find it themselves, even if they've never visited it before in their lives. So really a remarkable form of communication, and of course it's attracted substantial research interest over the decades since it was first described. But of course, the dance is not the only way that bees share information about foraging resources. They also are very adept at acquiring and learning about olfactory information. One way they do this is simply through antination. So you see here this bee is covered in pollen. Whatever flower it's been foraging on, it comes back to the hive smelling strongly of that flower. And other bees, by antinating this individual, can rapidly acquire and learn about the scent of whatever resources this bee has been foraging on, and potentially use that information to help them locate that site or similar sites themselves. Likewise, bees will often engage in trophallaxis. This is liquid food exchange or transfer. So how this works is a forager bee that's collected some nectar at a flower comes back to the hive, and it will purposely provide small samples of this nectar to other foragers who, by receiving this nectar, can learn about the scent, the taste, and potentially the quality of the resource this bee has been foraging on, and again, can help them potentially better locate similar or that exact resource themselves in the field. And of course, this question of whether bees primarily are relying on olfactory information or whether they're primarily using the dance to locate foraging sites was kind of central to the dance language controversy that played out in the 1960s and 70s between Carl von Fritsch and his students and Adrian Venner and his students, where Carl von Fritsch, of course, believed that bees could use the dance to locate food sites, whereas Adrian Venner thought that bee foraging could entirely be explained by olfactory information transfer, and that bees could not access the spatial information contained in the dance. And of course, we now know that, yes, bees absolutely can follow the instructions provided by the dance. But it's interesting is if you go back to the paper by James Gould in 1975, where he shows very clearly and once and for all that absolutely bees can follow the instructions provided by the dance. We also see that the extent to which bees rely on dance information is very much dependent on conditions within the hive. So without going into too many details of this experiment, what essentially Gould did was he tricked foragers into dancing for sites they'd never visited. So the dancers only ever visited this forager station, but he was able to cleverly make it seem like they were dancing for these other sites in the field. And he shows that he can manipulate the conditions in the hive such that sometimes bees are primarily following the instructions provided by the dancer, or they seemingly ignore the dance information and potentially are foraging entirely on the basis, or primarily on the basis of olfactory information that's being spread within the hive. So for my postdoctoral research, what I was interested in was trying to understand how do these different forms of interaction ultimately combine to help bees locate food. So what is the relative importance of waggle dances versus antennation versus trophallaxis, and how might this vary across different foraging contexts? But this is a challenging question to address because generally bees have access to all of these information sources simultaneously within the hive. So we need to find a means of being able to tease apart their relative contributions. And that's where multi-layer network analysis can potentially be of use. So to give you kind of a brief outline for where I'm going with this talk, first I'm going to briefly introduce network-based diffusion analysis, which is a technique for studying social transmission within animal groups. I'll then discuss an experiment where we employed network-based diffusion analysis to evaluate how important these different social transmission mechanisms are across the context of recruitment versus reactivation. And I'll get into more in a bit about what exactly those processes are. And then finally, I'm going to end by focusing in particular on the role of the dance during forager reactivation. Network-based diffusion analysis was a technique that was initially developed to help bring social learning research out of the lab and enable for the study of social transmission within wild animal populations or other populations where the animals are able to freely interact with one another. And the basic idea here is that let's say we have information about the order or timing at which a particular behavior is spreading through a population of individuals. So going back to our monkeys, perhaps we have information about the order in which individuals are first exhibiting or displaying a particular foraging technique. And let's say that we have a network that we believe reflects opportunities or predicts opportunities that individuals will learn from one another. If social transmission is involved in this behavior, then we would expect that the order in which individuals first exhibit this trait should map onto that network. And the more closely that it does so, the stronger the estimated importance of social transmission in the spread of this behavior. So we can use network-based diffusion analysis to do a couple things. We can use it to detect and quantify the relative importance of social transmission in promoting the spread of a particular behavior through a population. And if we have more than one network, we can use network-based diffusion analysis to evaluate which of those networks or which combination of networks best predicts the spread of our behavior of interest, which is what we did with our honeybees. So we trained bees to particular foraging stations, and so we observed the order in which bees located particular foraging sites. And meanwhile, we built up interaction networks based on interactions that went on in the hive between bees in terms of waggle dances and trophallaxis and antennation, and then use network-based diffusion analysis to evaluate how the relative importance of these networks varied across different foraging scenarios. So the first study that I want to share with you is an experiment where we used NBDA to evaluate how important these three networks are and how their contribution varied across two key foraging contexts, recruitment and reactivation. So again, we trained bees to artificial feeding stations. You can see one of those shown here with several bees foraging very happily around it. And we individually marked our foragers with paint so that we could tell them apart, so that we knew the foraging history of each of our each of the individuals we included in our experiments. We knew which feeders they knew of and which ones they had never visited before. And we looked at two key foraging contexts. We looked at recruitment. This is where bees are locating a novel foraging site they've never visited before, based at least partly on social information that they've received in the hive. And reactivation. This is where bees know about a particular foraging site. They've been foraging at it, but they're not currently foraging at it. Their foraging has been interrupted for some reason, most commonly because bees stop foraging overnight, so they then need to resume foraging in the morning, or when conditions are otherwise good, or other scenarios could be like a rainstorm sweeps through the area, everyone stops foraging during the rainstorm, but once the rainstorm passes, bees need to reactivate to resume foraging at these previously profitable sites. So what types of social information might bees use in the hive to help guide their reactivation decisions? So this is what our experiments generally look like. We have two groups of foragers from the same hive that we train to two different feeders. In this case, they were located 200 meters away from the colony. Each forager group only knows about its feeder. It's never visited the other feeder. And the day before the trial, we provided a scent at each of these feeders, and we used the same scent at both feeders. So what we did here is we mimic a scenario in which two groups of foragers from the same colony are foraging on the same species of flower, but at different unique foraging patches. Then on the morning of the trial, we took one of the feeders away. We left it empty. And what this does for us is it creates on demand a group of foragers who had been foraging at a site but are now no longer knowledgeable about a profitable foraging location. They are amenable to locating a new site, and meanwhile their nestmates are advertising this other site that they've never visited before but that smells similar to what they had been foraging on. So we look to see what is the order in which bees from our empty site, in what order do they locate this new to them full site? And meanwhile, we're recording all of the dance following, trophallaxis, and antination interactions between bees advertising this full site and their interactions with the bees that are searching for that site that have been trained to the empty feeder. At the same time, we also recorded the order in which bees trained to the still full feeder return to that feeder for the first time during the trial. So this is our measure of reactivation. So the order in which they first reactivate to their familiar feeding site. And again, we're going to examine which of our networks or which combination of networks best predicts the order in which they do so. So for this experiment, we had 4 colonies, and we focused on 56 recruitment events and 67 reactivation events. So first, for recruitment. What we have here on the x-axis is the percentage of recruitment events that the network-based diffusion analysis estimates we can attribute to each of our focal network types. And when we do this, we find, or the NBDA estimates that nearly all recruitment, about 97% of recruitment events, could be attributed to the dance following network alone. And that trophallaxis and antination explained basically no recruitment. So on the one hand, this makes sense. Recruitment is definitely the context we would have expected the dance to be especially important, because that's where the spatial information being provided by dancers would be especially helpful in being able to locate a site you've never been to before. But we had purposely set up our experiment, particularly using relatively short distances to the colony, in such a way that we believe that olfactory transmission would have been a viable route for some bees to follow, that they could use the knowledge that there was another site that smelled like what they've been foraging on to be able to search the surrounding area of the hive and locate this site even without the use of the dance, or in addition to the dance. Nevertheless, that did not seem to be the case, at least under the experimental conditions of our study. So the dance is absolutely key for recruitment. However, we also know that most dance followers are actually experienced foragers. They're not following the dance for the purpose of recruitment. And in fact, most bees might be following the dance primarily for reactivation. So again, this is where bees know about a particular site. They're not currently foraging at that site, but they may resume foraging based on social information they've received in the hive. So we see a lot of dance following during this context, but it's often very brief. It's unclear how important dance following is for reactivation, whether bees, for example, are paying attention to the spatial information provided by the dance. And this is also a context where we would expect olfactory transmission to be important, because simply knowing that a site that smells like what you've been foraging at is once more being collected from might be sufficient information for a bee to decide it will resume foraging at its site as well. So we carried out another network-based diffusion analysis, but this time looking at reactivation. What we find is a very different picture from recruitment. We find that dance following is still important. It accounts for about 18% or so of reactivation events, but that our other two networks, trophallaxis and especially antination, also are important here and can explain a substantial proportion of reactivation. So here we see that reactivation seems to be driven not just by dance following, but by a combination of all 3 network types. So we've seen how using a multi-network network-based diffusion analysis can help us disentangle the relative importance of different forms of transmission across different foraging contexts. We saw the dance was key for recruitment, whereas reactivation seems to be driven by a variety of interaction types. Next, we want to focus more on this context of reactivation, and especially how important the dance truly is for reactivation. So that's what we are going to discuss next. So as I mentioned before, in most cases when bees are following dances, oftentimes they already know about one or more sites in their environment. So this is from a 2005 study where they followed several bees across their foraging careers, and they estimated that out of all of the waggle dance following events they observed, only about 12 to 25% of dance following was for the purposes of locating a novel, never before visited site or for recruitment. The remaining 75% of dance following interactions appeared to occur in the context of reactivation. So scenarios where bees already know about one or more sites in their environment. So given that the dance is so prevalent in this context, I wanted to explore in more detail what is the role of dance following during reactivation? What types of information might bees be gaining from following the dance? Do they pay attention to the spatial information transmitted by the dance during this context? And again, how does this combine with other forms of interaction in the hive? So we carried out another follow-up experiment. Similar kind of setup. We've got two feeders. Though this time each of the feeders has a unique scent. So for example, one might be scented with lavender, whereas the other is scented with geranium. So bees, again, only knew about one of these sites. They'd only been visiting one of these sites. Though, again, there was a group of foragers at each of these sites. Then we took away our feeders, so they're empty. So we're simulating essentially like an interruption in their foraging. So imagine like a rainstorm swept through the area, no one can forage for these 4 hours. So the feeders were available, but they were empty. There was nothing for the bees to do, and gradually they kind of decreased how often they were coming. But still, sometimes they would inspect, which we relied upon, because then later in the afternoon, we replaced each of these feeders. They were once more full. They were providing the same scents they had been before. And then we recorded the order in which bees ultimately returned to resume foraging at their familiar feeding site. So first we wanted to examine how the networks of interactions during this reactivation process, whether they were structured according to the foraging sites being exploited by these foragers. So these are networks from one of our trials. Each node here is an individual forager. The colors correspond to the scents that were provided at the sites that these bees had been foraging at. And what we find is that dance following and antenation in particular were assorted according to feeder type. Or in other words, prior to their reactivation, bees interacted more frequently and they interacted for longer with nestmates that were advertising a site that was familiar to themselves. Or put another way, bees were able to organize themselves in such a way that they were more likely to be exposed to or have access to that information that's most relevant to them, that is, information about their familiar foraging site. We do see similar patterns in trophallaxis, but ultimately this pattern was not statistically significant. So we found that during reactivation, honeybee forager networks were organized according to foraging sites. So bees interacted more frequently and for longer with those bees that were advertising sites that were familiar to them. We next wanted to explore the rate of transmission of information through these networks. So we performed another network-based diffusion analysis. So as before, we're looking at the order in which bees first return to their familiar feeding site and the ability of the dance fouling, trophallaxis, and antenation networks to predict this ordering. But we're going to introduce a bit of added complexity here in that we're going to take each of those networks and partition them into subnetworks where connections either link together bees where there is a match between the information being provided by the informed individual and the recipient of that information, or where there's a mismatch. So for example, for the dance following network, in the same feeder scenario, the feeder that's being advertised by the dancer is the same feeder that the dance follower was familiar with and would ultimately reactivate to, whereas in the different feeder condition, the dancer is advertising a site that's unfamiliar to the dance follower. So the dance follower would reactivate to a different site than the one being advertised by the dance it's following. So On the x-axis here, we have the social transmission rate parameter. This is a parameter estimated by the network-based diffusion analysis, and it represents on a per bout basis the acceleratory effect of that bout of interaction over the likelihood that an individual will reactivate. And when we do this, we find that dances have a very strong impact on the likelihood of reactivation. So following a single bout of dancing has a much stronger impact on your likelihood of reactivation than engaging in a single bout of trophallaxis or antination. And this is especially the case if the dance indicates a site that's familiar to the dance follower. So even though bees already know about the location they're returning to, they still seem to be paying attention to an extent to the spatial information transmitted by the dance. And when that matches up with their own personal navigational memories, it makes it more likely that that bee is going to respond by reactivating. So whereas the social transmission rate parameter captures the importance on a per bout basis, we also then combine this with information on the strength of connections in the network to estimate the percentage of reactivation events that we could attribute to each of our networks. And when we do this, we find that overall, same feeder interactions explain substantially more reactivation events than different feeder interactions. And this is primarily due to the contribution of same feeder dance following networks. That the same feeder dance following network explains substantially more reactivations than the different feeder dance following network. Trophallaxis explained hardly no reactivation events, but antination once more explained a substantial proportion and actually a similar amount as each of the dance following networks. So whereas each individual bout of dance following had a very strong impact on the likelihood of reactivation, the fact that bees engaged in so many more antination bouts led to antination having a similar overall contribution in terms of how many reactivation events that network ultimately was responsible for. So to summarize, we found that honeybee in-hive interactions during reactivation were organized according to floral resources. So bees were more likely to interact with other bees that were advertising a site familiar to themselves. That dances, especially when those dances indicated familiar locations, were strong motivators of reactivation. And that overall, same feeder interactions drove the majority of reactivations in our study. All right, so let's pull this all together now. We've looked at the role of 3 different forms of interaction in transmitting foraging information, and we've looked at this across 2 foraging contexts, recruitment and reactivation. So as we saw, for the context of recruitment, this was primarily explained by the dance following network and the dance following network alone. Trophallaxis and antination didn't really explain much in the way of recruitment patterns. But dance following also is an important component of the reactivation process. In the language of system science, this is what is known as a pluripotent signal. It's a single signaling component that has or fulfills multiple functions simultaneously. And what this means is that even though relatively few individuals at any given time are using dance information for the purposes of recruitment, the cost of producing that spatial information that's so critical to recruitment is offset by the benefits that the colony is likely gaining from all of the bees that are still utilizing the dance for this function of reactivation. However, reactivation is also explained by not just dance following patterns, but patterns of antination and to a lesser extent trophallaxis. And this is what's referred to as a degenerate signal, or a case of multiple signaling components that overlap in their functionality, at least under some contexts. And this means that ultimately this can enhance the robustness of the collective foraging within the colony, as the decision to reactivate is not dependent solely on engaging in dance following or antination or trophallaxis alone, but it can be any of those interactions or any combination of them, or even, and I didn't really touch on this before, none of them at all. So we did see evidence of also spontaneous reactivation as well. All right, so some quick takeaways. We've seen how employing a multi-network NBDA can be a useful tool for evaluating how different forms of social interaction combine to shape social transmission processes. We've seen how we can employ this technique to evaluate the relative contribution of different forms of interaction in the honeybee hive and how their importance varies across foraging contexts. And finally, I think one of the values, from at least for me, of employing these types of multi-layer, multi-network approaches is that they can facilitate a systems approach to thinking about the way in which the functionality of social behaviors is influenced by the way in which they diverge and overlap in their functionality across contexts, and the way that this influences the robustness, efficiency, or evolvability of social processes. And so finally, with that, thank you for listening. Thank you as well to my co-authors on this work, Ellie Ledbeater and Will Hoppett, as well as all of those who helped me with the feeder experiments. Here's my contact info. If you have any questions or you want to reach out, I'd love to hear from you. Thank you so much.